Now, as part of the antigen presentation process, before the antigens can be presented and activate the adaptive immune system, they have to first be recognized by a T lymphocyte. Remember, T cells or T lymphocyte cells are one of the cells of the innate immune system. So the way this happens is macrophages and dendritic cells, also part of the innate system, will be the first to bind to the antigen, to recognize and bind to the antigen on the foreign material. They will then present the antigen to a T cell for what we call recognition. In other words, for recognition as not belonging in the body. It's the T cell's job then to release a chemical called a lymphokine. And these lymphokines, like the cytokines we've talked about, will draw other immune cells into the area, including the cells that make up the adaptive immune system. And by drawing them or attracting them into the area, those cells will be activated. Now, I have a note on the slide about the terrible mechanism of human immunodeficiency virus because it involves T lymphocytes. You remember what I just said. The T lymphocyte is a critical part of the process of the activation of adaptive immunity, and it requires T lymphocytes in order to happen. And what the human immunodeficiency virus targets are T lymphocytes. The human immunodeficiency virus, HIV, infects T lymphocytes and destroys them. So a person who has HIV infection and then goes on to develop AIDS is a person who does not have a functioning immune system. And the reason AIDS used to kill you is because you cannot survive without a functioning immune system. Luckily, now in 2020, we have really good drugs to help people who are infected with HIV so that they never go on to develop AIDS. They never get to the point where their immune system is destroyed. Now, the response to this antigen presentation process occurs in two ways. The first is referred to as cell-mediated immunity, and the second is antibody-mediated immunity. And we're going to talk about both of these separately. So this is the side of the immune system that is referred to as the adaptive immune system. Now, cell-mediated immunity is the adaptive response that doesn't involve antibodies. Instead, it involves specific types of white blood cells that recognize each antigen separately. So now we're into the side of the immune system that is specific. In other words, it's not just attacking and destroying general foreign material, but rather identifying specific antigens and responding to them. So there are a particular type of T lymphocyte that is involved here that's referred to as a cytotoxic T lymphocyte. And this is another type of phagocyte. This is a cell that is capable of performing phagocytosis, taking in or eating this foreign material to break it down. And just like we saw on the innate side of the immune system, these cells will release a variety of these immune chemicals, cytokines again, and lymphokines, in response to coming into contact with this antigen. Now, antibody-mediated immunity also goes by another term. The older term is humoral immunity, but you will still see the antibody-mediated immunity referred to as humoral immunity. When they're stimulated, B lymphocytes, or what we sometimes call B cells, become activated. And what happens is they go through a process of maturation or differentiation, and they become two very particular types of immune cells called plasma cells and memory cells. So the B lymphocyte has to get activated in order to mature and divide and produce plasma cells and memory cells. Plasma cells are the cells that produce the antibodies, and memory cells are going to preserve the memory of the antigen that was presented. What that will allow for is the next time that antigen gets into the body, it will result in a faster immune reaction to that antigen. On this slide, we can see a drawing of this process that ends up producing memory cells and plasma cells. Up at the top, you can see a representation of a B cell or a B lymphocyte. The B cell got its name because of the term bursa. That's just a little bit of trivia for you. The B cell is one of the cells in the immune system that has an antibody protruding from its surface. Now, we haven't spoken about antibodies yet, but antibodies are what make this so specific, because each individual antibody is only going to bind to one particular type of antigen. So again, this is a component of this antigen presentation process. The B cell gets a presentation of antigen, and that presentation is being done by a T cell. In the drawing, it's labeled as a Cd4 activated T-cell. Remember, the T-cell is a critical part of antigen presentation, and it involves the secretion of these chemicals called cytokines. So the B cell gets activated, and the B cell begins to differentiate. It begins to mature or grow up and change. It will then divide to produce two different types of cells. One is the memory cell, and one is the plasma cell. The plasma cell will then go on to produce antibodies, and that's what these are. The memory cell holds on to the memory of this antigen. So the next time that antigen gets into the body, the memory cell will very quickly become activated, and it will allow for a faster, specific antibody-based response to that antigen. So let's take a few minutes and talk about antibodies. It's important to know that the other term that you will hear for antibody is immunoglobin or immunoglobin. Those two things mean the same thing. Antibodies, as we've just said, are produced by a type of cell called a plasma cell, and plasma cells come from activated B cells. Antibodies are simply specialized proteins, and as you've seen in the drawings, they have a very particular shape. They look like the letter Y, and they literally do look like the letter Y under a microscope. So just like the letter Y, they have this base, or what we call a stem, and two arms. Now, the arms are often referred to with the abbreviation FAB, and that means this is the antibody binding region of the antibody. So the antigen binds right into that V shape in that Y. The stem is referred to as FC, with C standing for constant. So at the very bottom of the stem of the letter Y, this is the place on an antibody that allows it to bind to the surface of certain immune cells, because there are immune cells, including B cells, that have antibodies jutting off of their surface. The other thing that you will hear an antibody described as having are different types of proteins based on weight, if you will. So antibodies are often described as having 2 heavy chains in them and 2 light chains in them. Don't worry too much about this language. I just wanted to mention it to you because you'll often see those terms, heavy chain and light chain, in descriptions of antibodies. And I'll show you these in just a minute. Antibodies are going to circulate in the bloodstream, and they're also going to circulate in the lymphatic system. Remember, antibodies are specific. They are going to be able to bind to a very particular antigen. In fact, a very particular epitope on a particular antigen. And that specific antigen is the one that stimulated the B cell that ended up producing the plasma cell that produced the antibodies. So antibodies are part of the adaptive side of the immune system, the specific side of the immune system. Now, some other language that we need to be aware of is this language that describes an antibody as either being monoclonal or polyclonal. The term monoclonal translates as one clone, and we define it as being produced by a single plasma cell. So a monoclonal antibody is an antibody that gets produced by a single plasma cell. In other words, all of the antibodies that come out of one plasma cell would be called monoclonal antibodies. They are all identical. They're all clones of each other. Polyclonal antibodies are different. Polyclonal antibodies are all produced by different plasma cells, and so they often respond to different epitopes of the same antigen. So monoclonal, one type of clone; polyclonal, many types of clones. The monoclonal type of antibody, all of those antibodies that are monoclonal respond to the same epitope on an antigen. But a polyclonal type of antibody includes a group of antibodies that each respond to a different epitope on an antigen. Now let's take a minute and look at the different types of antibodies. There are 5 classes of antibody, and these are referred to as IgM, IgG, IgA, IgD, and IgE. The Ig part of the name stands for immunoglobin, which, if you recall, is just another term for antibody. So there are a couple things for each type of antibody that I want us to be familiar with. For IgM, you should know that this antibody exists exclusively in the bloodstream, so you're not going to find it in other locations in the body. This is an antibody that cannot cross through the membrane of a cell. This is the type of antibody that is responsible for transfusion reactions. A transfusion reaction, if you're not familiar, is the kind of immune response that occurs when a person receives the wrong type of blood in a blood transfusion. IgG is quite different. IgG is found in the blood and in various secretions throughout the body. IgG is capable of crossing the placenta. And so it's IgG that gives a newborn baby immunity from its mother. The IgG in the mother crosses the placenta and enters the fetus's body. And those antibodies, those IgG antibodies, will then circulate in the newborn baby for several weeks and will give that baby a passive type of immunity. The baby didn't make the antibody. It got the antibody through the placenta. But the antibody will function in the baby's body just like it would in the mother's body. Now, there's been a lot of talk about antibodies in the news recently with regard to COVID-19. If you've been watching news stories, you know that we are collecting blood from people who have recovered from COVID-19 and who have mounted an adaptive immune response and produced antibodies against this virus. By harvesting the serum in the blood, the portion of the blood that doesn't have the red blood cells in it and the white blood cells in it, but has the antibodies, by harvesting that, we are hoping that we can use that as a treatment. We can give that to people who are really suffering from COVID-19, who are close to death, and provide them with passive immunity. Give them someone else's antibodies and allow those antibodies to destroy the virus. There's a lot of hope right now that antibody treatment may help save some of the sickest COVID-19 patients. Now, there's some other language on this slide that we need to talk about, and that's this concept of primary versus secondary antibody, and I will get to that in just a minute. IgM is the most prominent type of primary antibody. IgG is the most prominent type of secondary antibody. And again, we'll get to that in just a minute. IgA is the type of antibody that really provides protection at surfaces, at body surfaces. So we find IgA in places in the body that are hollow and again lined with epithelium. IgA is very prominent in respiratory secretions and in saliva and in the secretions of the gastrointestinal tract. There's IgA in your tears, and in the secretions in the urinary tract and in the reproductive system. There's a lot of IgA in breast milk, especially in colostrum. Very, very important type of antibody. The IgD antibody is quite unique in that it only functions attached to B cells. So you won't find IgD circulating on its own. All of the IgD is attached to B cells at the stem, remember the Fc. So a very different type of antibody. And finally, there's IgE, which we do find in small amounts in the bloodstream, but we also find it in other parts of the tissue. IgE is an important antibody primarily because it mediates what are called hypersensitivity reactions or allergic reactions. So IgE is the type of antibody that is responsible for an anaphylactic reaction. So let's take a look at some drawings of antibodies and think a little bit more about their structure. What you can see on this slide is a drawing of an IgG, an IgM, IgA, IgE, and IgD. Now the first thing you notice is that Y shape. And of course, all of them have this Y shape in some form or another. The next thing you notice is that some antibodies have more than one Y in them. The IgM, for example, is actually made of 1, 2, 3, 4, 5 different Ys. And the IgA is made of 2 that are connected to each other. The next thing to notice is the heavy chain and light chain language that I mentioned. So if you look at the Y in the drawings, you can see that it's actually composed of 2 structures, 2 parallel structures. Remember, we call this the FC, the constant region. We call these the FAB. This is where the antigen binds. The light chains are the outer strands, if you will, on the FAB. The heavy chains are the 2 remaining strands, if you will, on this molecule. Okay? So the heavy chains are longer than the light chains. It's all protein. It's all amino acids. It's just the way it's structured that we refer to as heavy chains and light chains. Now, the other thing I want you to notice is that even in the case of IgM and IgA, where you see multiple formations of Y, it's always the FAB region that is sort of pointing out to the environment. So this is where the antigen binds here. This is where the antigen binds here. Here is where antigen binds. Down here is where antigen binds. Okay, it's the bottom part of the stem of the antibody, particularly on IgD. This is the one, if you recall, which is only found bound to B cells. And this is the location that binds to the B cell. Now, on this slide, this is just another drawing of an antibody. It's one, so it's a little bit easier to see the structure of it. This represents an IgG. You can see in this turquoise color, this is a heavy chain, and this is a heavy chain. And in this sort of fuchsia color, this is a light chain, and this is a light chain. And this area here and this area here are referred to as FAB, because this is where we expect to see the antigen, on this end of the molecule. And down here, this is the FC region. Actually, this whole stem is the FC region, or the constant region. Now, the last thing I want to talk about today, I referred to earlier when we were talking about types of antibodies. And I mentioned the terms primary and secondary antibodies. IgM were the most common primary antibodies, IgG the most common secondary antibodies. Now, this is not something I want you to be too concerned with. You don't need to know the details of this, other than to know that that terminology is used for a very common type of assay that's done in medicine called an ELISA. ELISA stands for Enzyme-Linked Immunosorbent Assay. And if you go into the healthcare field, you will encounter ELISA assays being done in laboratories. So there are different types of ELISA tests. And again, we're not going to have the opportunity in this course to talk about ELISA technology in general, but we can speak about what are called indirect ELISA tests that involved primary and secondary antibodies. So take a look at the diagram. This represents the process that occurs in an indirect ELISA test. So this would be a test that you would use if you wanted to know if a person had an antibody present in their blood against a particular pathogen. So in the test, there's a kit that comes with antigen specifically bound to the bottom of wells inside a plastic plate. So the antigen is there, and we're going to test to see if there's an antibody to that antigen in the person's blood. So we add the person's blood in, and we look to see whether or not antibody binds to antigen, essentially. This would be what we would call primary antibody. Antibody that binds directly to antigen. And that's what they're showing us. You can see an antibody binding here and an antibody binding here. And then they're telling us that any antibodies that don't bind get washed off. They get washed away during this testing process. Now here's where the secondary antibody comes in. And the secondary antibody is actually provided in the test kit. It's an antibody that's going to tell us if there's other antibodies present. It's an antibody that will bind not to the antigen, but to another antibody. And that's what they're showing us in the picture. So if there's antibody present in the sample, it will stick to the antigen that is present in the test kit, that is stuck to the bottom of the well in the test kit. And then by adding the secondary antibody, it will bind anywhere that there is patient antibody bound. And what makes secondary antibody especially unique in an ELISA test is that it is stuck to an enzyme. So they build these secondary antibodies to have an enzyme attached to them. And in the last part of the test, we add a substrate molecule into the test wells, and we allow the enzyme to act on that substrate and turn it into a product. And it's a product that we can visualize, that we can see. It creates a color change typically. So what this indirect ELISA will tell us is, are there antibodies present, primary antibodies that bind to antigen? Are they present in the sample? And if they are there, how many of them are there? We can use this test to quantify antibodies or to determine a titer. So again, we don't have the opportunity in this course to talk about ELISA technology in any depth, but it is a very important type of technology in medicine. It helps us in medical testing quite a bit.